for examining video number 6, I did the developmental network, which is the second topic. So this presentation, these will represent dragons with spikes, and these will represent the dragons without spikes. So for the wild type sequence, spikes will be produced and shown, as you can see from this dragon. That is because the S activates the P. The P then represses the I, so the I cannot repress the K. So the K is free to activate the E, and the E is free to activate the spikes. In the P- strand, spikes are not produced, as you can see from this little dragon here. And that is because there is no P to repress the I, so the I can repress the K, blocking the E from being activated and the spikes from being activated. In the I- strand, the dragon, the spikes will be shown, as you can see here, and that is because there is no I to repress the K, so the K is free to activate the E, and the E is free to activate the spikes. In the I- and E- strand, the spikes will not be present, as you can see here from our little dragon guy, and that is because there is no E to activate the spikes. In the S- and K- strand, the spikes are not produced. It's because there is no K to activate the E, so the E cannot activate the spikes. So transcription factors are proteins that help turn specific genes on or off by binding to nearby DNA. So a repressor represses transcription, getting in the way of transcription factors, the RNA polymerase. So as you can see here, here's the binding site for this repressor. The repressor comes and binds, and it blocks transcription factors and the RNA polymerase from binding to it, preventing further transcription. So when you have the same genome, it is possible to show different phenotypes despite having the same genome. And that is because you need silencers. Silencers are faraway clusters of binding sites for repressors, and they bind certain transcription factors. So mutations only occur in these silencers, not in the whole genome, because if they were to occur in the genome, then every single cell would be affected, but when they only occur in the silencer, only that specific cell is affected. So for example, with our dragon example, if our mutation occurred in the genome, then the dragon would not have spikes, but because it only occurs in the silencer region, only certain cells do not have the spike phenotype. So here's an illustration of that. So here's our normal wild type sequence with the spikes being produced. But the repressor will come and will bind to the K. Repressing the K is the transcription factor binding site. So it will repress K, blocking the transcription factor from coming to bind to K. So the K is no longer available, so no spikes will be produced. And that prevents spikes from the spike phenotype in certain cells, even though it will be produced in the same type of cells with the same genotype. Here are my references.